In this talk, we're going to learn about the fertilization and embryogenesis that occurs in angiosperms. By the end of this talk, students should be able to describe the different types of pollination and the agents that perform pollination. Students should also be able to explain the steps involved in fertilization and embryogenesis, and finally be able to identify the different parts of a dicot seed. Now that we've gone through the process of megagametogenesis and microgametogenesis, let us look at how the male gametes and female gametes are able to meet each other. And this is where pollination comes into play. The process of transferring the pollen grains which harbor the male gametes from the anther to the carpel is pollination. Now, there are two main types of pollination, and so self-pollination is where you can have the transfer of pollen from the anther of one flower to the stigma of the same flower. And remember, the stigma is the part of the carpel, and that is the place where the anther is actually going to be deposited. So in another scenario of self-pollination, you can have the pollen from the anther of one flower be deposited on the stigma of a flower that's present on the same plant. And so that way both flowers are present on that same plant, and you're having the transfer of pollen from one flower to another flower that belongs to the same plant. Another process of pollination is cross-pollination. In cross-pollination, we are also having transfer of pollen from one flower to another flower. However, that second flower, which is getting the pollen, it is present on another plant that belongs to the same species. Now, this transfer of pollen from one flower to another flower is usually done with the help of agents. Pollination can be mediated by different agents that can be abiotic or biotic. The abiotic agents tend to be primarily wind and water. Now, we had seen how wind can help in pollination in the case of gymnosperms, and they can also help in pollination in angiosperms. So in the figure right here, we can see water playing a role in pollination. And in this middle figure, we can actually see how wind can play a role in pollination. In addition to abiotic factors, you can have biotic agents also, which are different types of animals. And when I say animals, I mean they can be insects or invertebrates. And normally we are all familiar with the butterflies and the honeybees, but they're not the only ones that can be promoting pollination. You have many other types of insects also that promote pollination, which include ants, certain types of worms, so on and so forth. Invertebrates are not the only creatures that can help in pollination, but even vertebrates can help in pollination. So naturally, pollination can also be mediated by birds like hummingbirds, by mammals like bats, or by reptiles like some sorts of lizards. Thus, there is a wide variety of biotic agents that can mediate pollination, and plants have come up with all kinds of adaptations to attract all these organisms to enable them to go through pollination. Now, once pollination has occurred, the pollen grain binds to the stigma of a carpel. Now, first compatibility is checked. So if the pollen is compatible with the stigma, that's when it's allowed the process goes further, where the pollen gets hydrated. This hydration allows the exine to degrade, and the cytoplasm of the vegetative cell bulges out through an opening called as the germ pore. Now, we had looked at the germ pore before, and we saw that monocots normally have one germ pore, whereas dicots tend to have 3 germ pores. As you have this tube coming out through this germ pore, that's where the pollen tube is coming through. The tube nucleus, if you remember from previous slides, the pollen grain has the vegetative nucleus, and it also has the 2 male gametes. So the tube nucleus, which is another name for the vegetative nucleus, is present at the apex of the pollen tube, which is what we can see in the blue box. The male gametes, on the other hand, are right behind that tube nucleus as the pollen tube grows. The pollen tube grows in length, and as we can see in the blue box, we can see how the pollen tube is getting longer and longer. So just to remind you, the pollen grain is actually on the stigma of the carpel. And as the pollen tube gets longer and longer, as we see in the figure, it passes through the style and it finally reaches the ovary and enters the ovule. So the pollen tube is able to go through the style by following a chemical gradient. And then when it enters the ovule, which is shown in that small box, through the micropyle. Once it enters the ovule, the pollen tube is guided by the synergids so that it can actually go to the egg. So remember, the synergids in the egg are the 3 cells that are near the micropyle of the ovule. Now, once the pollen tube is able to enter into the ovule and is guided by the synergids towards the egg, the male gametes can now be released. And so these male gametes, when they are released, there are 2 of them, and one of them is going to fuse with the egg cell. And this is what results in the formation of the zygote. The other male gamete fuses with the definitive nucleus, which already has 2 nuclei. So after this process of fertilization, and in this case there have been 2 fusing events. So we formed the diploid zygote through the fusion of the egg and the sperm cell. And so this way we had 1 fertilization event that resulted in the diploid zygote. But we also have a second fusion event where the male gamete fused with the definitive nucleus, which is diploid. This results into this process is called double fertilization, where you're having these 2 fusion events. The formation of the male gamete fusing with the definitive nucleus gives rise to a triploid cell, which is called as the endosperm. So in this case, we can see the zygote is diploid and we have a triploid endosperm. Once fertilization occurs and the zygote is formed, which is shown right here in the blue box, embryogenesis begins. And in the process of embryogenesis, that fertilized egg is going to go through different stages and ultimately is going to form a mature embryo, which is what is shown to the far right. This mature embryo is then packaged into this coat, it's called, and forms a seed. So inside the seed, we have the embryo, which is covered by a seed coat. So we should remember that all of this is happening in the ovule region. And so sometimes when you see multiple seeds present, say like in a fruit, then those multiple seeds have basically arisen from all the different ovules that got fertilized. Now that double fertilization is complete, we saw that the double fertilization resulted in an endosperm that provides nutrition to a diploid zygote, which undergoes development to form an embryo. Now, the first stage of development for the zygote is it undergoes asymmetrical cell division and gives rise to an apical cell and a basal cell. So in this figure where it says terminal cell, that upper cell is also called as the apical cell, and the lower cell is called as the basal cell. The apical cell is the one that's going to form most of the plant embryo, while the basal cell forms the structure called as a hypophysis and suspensor, and we'll see the roles of these parts in a bit. As the embryo undergoes cell division, so here we have a 2-cell embryo along with a suspensor that's being formed, which ultimately is going to form the 8-cell stage. So in this 8-cell stage of the embryo can be divided into 4 main domains. So in these 4 domains, we can also see those 4 domains in the figure that's in the box. So let us look at more details of what happens in these 4 domains. So in this 8-cell stage that we can see right here, we're going to look at the different domains that are being formed, and the portion of the figure in that blue box is what has been magnified in the figure that is there to the right. So the first domain is called as the apical embryo domain, and this is the part of the embryo that's going to have the shoot apical meristem and the cotyledons. The next part or domain is the central embryo domain. And this is the part that's going to have the hypocotyle, the root apical meristem, and parts of the cotyledons. The hypophysis is one that's below the central embryo domain, and the hypophysis can also be sometimes called as the basal embryo domain. It is the one that forms the radicle and the root cap. Ultimately, the suspensor is the one that is going to anchor the embryo to the endosperm. And the endosperm is what is going to provide nourishment to this developing embryo. And so the suspensor acts as a connection where the embryo is able to get its nutrition from the endosperm. To continue the process of embryogenesis, we come all the way to this 8-cell stage where we're able to see those 4 domains. Then we come to the 16-cell stage. And in the 16-cell stage, the embryo has more number of cells, but one of the key features is the formation of a structure called as the protoderm. And the protoderm is a meristematic tissue that forms the epidermis, or one of these outer layers of the plant. Now, after the 16-cell stage, we have a stage that's called as the globular stage. And in this stage, in addition to the protoderm that we can see in the figure in the blue box, we also have the formation of what's called as the procambium and the ground meristem. Now, the procambium is the one that will ultimately give rise to xylem and phloem. And these are those connective conduits, or those tube-like structures that helps the transport of substances in vascular plants. The ground meristem is going to form the ground tissue as that plant matures. This is a picture of the globular stage where we can see not only the head portion that was shown in the colored picture before, but we can also see the entire structure, including the suspensor region. After the globular stage comes the heart stage, and in the heart stage, this is where cotyledons are beginning to form. In the case of dicot plants, 2 cotyledons will be formed, and that's what we're looking at here. And these 2 cotyledons tend to form this heart-shaped structure. So once again, in this picture, we're looking only at the head, but when we look at the picture to the right, we can actually see the entire structure of that embryo. So here we have the heart stage that we just looked at, and this heart stage progresses to form the torpedo stage. In the torpedo stage, there's continuous growth of the cotyledons and there is axis elongation of that embryo. But when we look at that suspensor region, it actually becomes a lot shorter. Finally, the embryo undergoes maturation processes where embryogenesis is complete. And now what has to happen is this embryo has to be packaged in its seed, and the seed has to undergo desiccation. So it basically has to dry up. And this results in the embryo becoming dormant. Now, a lot of these processes like desiccation of the seed and dormancy in the embryo, they're all regulated by many different molecules. And one of the hormones called abscisic acid is the one that plays a really important role in inducing and maintaining dormancy of the embryo. It should be noted that this dormancy step is really important because it allows the embryo to remain as an embryo and not germinate to form an actual plant when the conditions are not right. So with this, we come to the end of embryogenesis. Now in this picture, it's basically showing the whole process of embryogenesis, but we're looking at mainly 2 parts. So we're looking at the structures formed by that basal cell, which are shown in purple, and the structures formed from the apical cell that are shown in yellow. So if you remember, the zygote, it undergoes mitosis to form an apical cell and a basal cell. So here's during the 4-cell stage, and we can see that we have that long suspensor that's formed from the basal cells, and we can see the apical cells undergoing cell division to form like the different domains. And here we have the globular stage where you should have differentiation of the cells. There we can see the process of formation of the cotyledons. And so even in this stage, when we look at that dark pink suspensor, it is quite long. And the part that is up that's arisen from that apical cell, we can see the growth of the cotyledons. And as development continues, we now see a really good heart-shaped structure. And now when we look at this stage, we are seeing more and more of the cotyledons growing. However, now look at that suspensor region. It is actually becoming shorter and shorter. And so when we look at the next region where we're coming to the torpedo stage, that suspensor length has really shortened, and you basically it's there's not that many cells in that suspensor anymore. And one of the reasons that this is happening is in this kind of embryogenesis, the suspensor is taking nutrition from the endosperm. But as that embryo is developing, that endosperm is getting depleted, and hence there's really not much of a function for that suspensor anymore. And so the embryo that has to be cramped into the seed, it wants as much space as possible, and hence the degeneration of the suspensor has to occur to allow for space to that growing embryo. Just to take a look at what is actually happening with that endosperm during embryogenesis, here we can see in this figure the dark green structure is the zygote, and the light green structure on top is that triploid endosperm. Now, when we go through embryogenesis, here we can see the 8-cell stage of that developing embryo, and the basal apparatus is shown in purple, whereas the apical cells are actually shown in green. And look at that endosperm. It is undergoing cell division to form a multicellular structure. And as an embryo is developing, you also have plenty of cells in the endosperm that are nourishing that embryo. Here that embryo is more of like a globular stage. And now we have the heart-shaped embryo, and there's still plenty of endosperm out there. But now as those cotyledons are growing and becoming bigger and bigger, we can now see that the endosperm is actually getting smaller. And so the suspensor is also starting to get smaller because there's really not much of a function for it. Ultimately, there's really very less of the endosperm left, and hence, once again, the need for that suspensor starts diminishing, and hence it starts undergoing degeneration. So now when embryogenesis is complete, we actually end up with the seed. So when we look at different parts of the seed, the outermost structure is the seed coat. And the seed coat protects the seed from different environmental factors, and the seed coat is actually formed from the integuments that cover that ovule. There are normally 2 layers to a seed coat, and the outer layer is called as the testa, and the inner layer is called as the tegumen. When we look at the different parts of the embryo in the seed right now, we can see the embryonic root that's called as the radicle, which is shown in the figure in the blue box, and we can also identify the embryonic shoot that's called as the plumule. Now, in addition to the radicle and the plumule, we have the cotyledons. And the cotyledons, in the case of dicots, we see 2 of them, but in the case of the monocot, there'll be only 1 cotyledon. Now, the cotyledon a lot of times can also be called as the false leaves or the first leaves of the seedling when that seed germinates to form a seedling. In addition to all these structures, we also have the epicotyle of the seed, and this is basically the base of the plumule. So the plumule is the one that's going to form the shoot, and right at the base of that shoot, as you can see in the figure, is the epicotyle. We also have another structure called as the hypocotyle, and the hypocotyle is basically the base of the radicle. So the radicle is the part of the embryo that's going to form the root, and so it's the tip of that root. And so when you go more further up, you'll be able to find the hypocotyle, which is called as the base of the radicle. To summarize, we learned about the processes of pollination and the steps involved in fertilization and embryogenesis. We also learned about the different parts of a seed that is formed from dicotyledonous angiosperms.